Batrachochytrium salamandivorans, or Bsal. This is the salamander-eating fungus that was first discovered in 2013 after it had almost entirely killed off several populations of fire salamanders in Europe. Researchers believe it spread there from Asia via the pet trade. And now they have yet to detect it in North America, but are very worried about its impacts if it arrives. The US is home to more salamander species than any other country, many of which belong to families that are known to be particularly susceptible to the disease. And now biologists are racing to figure out how different species react to Bsal in an effort to know how it may spread and where best to target conservation efforts. My name is Mike DiGirolamo, your host for Mongabay Explorers, a special podcast series about some of the most important recent reporting from Mongabay.com's global team. Join me for a deeper discussion of one such project, where I'll explore the issues with experts on the front lines of the looming salamander pandemic to find out what we know now and what is being done to keep North America's and the world's salamanders safe. Today, my guest is Dr. Karen Lips of the University of Maryland, College Park. She's a professor of biology and one of the most influential scientists in the world when it comes to amphibian infectious diseases. In 2014, she co-authored a New York Times op-ed urging our nation to take action on Bsal. Today, she's going to answer some of our most crucial questions on this Bsal pandemic, what we can do to keep the salamanders safe, and what this pandemic may mean for us.
Bsal is what we call a chytrid fungus with the official name Batrachochytrium salamandivorans, and it is dangerous to salamanders because it is a pathogen, it's a disease of salamanders, and it is highly lethal. So it can kill off many different species of salamanders very quickly. And then BD is the original, as we call it, frog chytrid.
BD is the culprit of an amphibian pandemic closely related to Bsal, which stretches back 25 years.
BD and Bsal are two types of chytrid pathogens. These are fungi that attack amphibians. And BD, Batrachochytrium dendrobatidis, was the one we discovered first, and it has now known to infect all types of amphibians: frogs, toads, salamanders, caecilians. More recently, we've discovered Bcell, and originally we thought this was primarily a disease of salamanders, and it does seem to affect salamanders more so than frogs and toads, but we now know that some frogs and toads can also transmit or carry the salamander chytrid as well. So these are both terrible diseases of amphibians that cause death, big die-offs in some cases, population declines, and in some cases even, we have seen extinction of species.
And when did you discover BD initially?
BD was discovered by 3 different teams back in the 1990s. So I was in a jungle in Panama and came across a bunch of dead frogs, and they were shown later to be infected with what we would call BD, and that was 1996. At about the same time, the Australians working in their rainforest had discovered big die-offs, population declines of their amphibians, and they had actually gotten a culture of this thing that was attacking the frogs and were doing experiments and showing that it was killing off the frogs. And the third team was here in Washington, D.C. at the National Zoo, and they had found that there was this crazy skin disease that they identified as a fungus that was wiping out the captive frogs at the National Zoo. And so we all got together around 1997-98 and compared the 3 sets of data and samples and concluded that it was the same thing in 3 different places.
And can you just articulate for our listeners the specific disease that they're catching is chytridiomycosis, correct?
Yes. Chytridiomycosis is what happens when an amphibian gets infected with either BD or with Bsal.
And can you tell us just a little bit more about that? What are the symptoms like? How does it affect the amphibian? and ultimately lead to their death.
And chytridiomycosis, it has a range of symptoms. In the worst cases, if you take an amphibian, a frog, like the golden frog from Panama, and you infect it with BD, they are very susceptible and they get infected very quickly. The infection grows very quickly, and they may die in a period of 1 or 2 weeks. Some of the symptoms that you see, they're not always consistent. Sometimes you see red discoloration, like bloody little hands and feet, where the infection is sort of disrupting their skin where it touches the ground. They'll be shedding skin on their body a little bit more frequently. A lot of times the animals it seems like they're uncomfortable and they sort of stick their butts up in the air. They don't seem to want to touch anything. I've noticed in the field that many of the animals that are very sick, their color is off, they're sluggish, they're not really responding very quickly. They may be out at a different time. Frogs generally are out at night, and we would find them out during the day. They'd be on the ground. They weren't able to move very well. So the symptoms range quite a bit among different species and with the level of infection. With Bsal, the salamander chytrid, the infection also at very high levels can be pretty traumatic on the animals, and they, again, get lethargic. They don't seem to be moving very much. And at some point, they kind of fade away. But this is not something where if you were just going for a hike in the woods and you found a dead animal that you would not be able to tell right off the bat if it was infected with chytrid or if it had died with chytrid. We often have to send these animals that we find to a lab to have tests done to confirm if they were actually infected. So it's not a super easy thing to diagnose for anybody.
Much like our current problem with COVID-19, animals are able to walk around with the fungus without displaying any symptoms at all.
Yes, given our new lingo in a pandemic world, asymptomatic would be a good description. Or it may be that the symptoms that they have are subtle enough or light enough that we might not be able to say that it's chytrid versus any number of other things.
Unfortunately, also like COVID-19, we aren't sure exactly the full extent of the damage the fungus can have on the animal, or even how it kills it.
I think we know a lot more about BD than we know about Bsal. And with BD, the infection in an amphibian we describe it kind of like a lawn. So they get infected and BD will fill the entire skin in really bad cases. And so if you looked at the skin under a microscope, what you would see is that BD is filling the skin. There's almost more fungi cells in the skin of the frog than there are frog skin cells. It physically disrupts the skin. And for many amphibians, they use their skin to breathe. They get their oxygen not from lungs, but primarily across their skin. And when this fungus disrupts the skin, they have a much harder time getting enough oxygen and getting rid of CO2. They also use their skin to pick up water, and when their skin is physically disrupted by the infection, they have a hard time getting enough water as well. The second thing that the fungus does to a frog in the case of BD is that the fungus is actually able to get into the immunological system of the frog and affect the immune response. That is, it's able to kind of turn off or slow down certain parts of the immune system that should be fighting off this infection. So lymphocytes are one of the ways that they use to fight off an infection. And when they have a very big infection, the fungus is able to slow down the production of those lymphocytes. So the frog, even though it's trying to fight it off, can't do it. In the case of Bsal, I'm not sure that we totally know yet exactly how it kills because the infection in the skin is a little bit different. If you think of BD as a lawn, basically coating the entire skin of a frog, Bsal is very different. It's more like pimples or chickenpox, where you have localized infections. So it's not completely covering the animal, but you see these little sort of blisters or pimples where the infection is really high. Sometimes you can actually see these little round circles on them, even with your naked eye. but in a lot of cases you might not recognize it as such. And so because it's not completely coating the skin of an animal, we're not exactly sure yet how it might be killing the animal. So it may still be messing around with the immune system like BD does, or it may have some additional tools in its box to kill off those salamanders.
However, BD has provided a baseline of research that scientists are using to inform their decisions on Bsal.
Certainly, we've had, 25 plus years, dozens, if not hundreds of people studying BD all around the world. And what we've done is sort of taken that knowledge and sort of used that as, well, this is probably how Bsal works. But of course, as we get more and more into it, we're realizing that, yeah, there's a lot of similarities, but there's certainly some differences as well. And we've just started really in the past couple years with Bsal. So we've got a long way to go to really understand what is Bsal, where does it come from, how does it operate in a frog, what does it do in the wild. So there's obviously a lot more work to be done.
And that is proving to be a challenge in and of itself. Despite the length of time that BD and Bsal have been spotted, scientists still aren't sure exactly where the fungus originated specifically, or even why it exists.
Bcell, we currently believe, came from somewhere in Asia. I think right now the closest locality we know of is somewhere in perhaps Korea or northern China, somewhere in that region. It's a little hard to know because getting into that area and sampling is quite difficult, as you might imagine. But as far as we know, both Bcell and BD are coming out of that same general region.
Is it possible that these fungal pandemics are being somehow created by human activity or interactions with the environment of people increasing biodiversity loss with deforestation and putting toxins in the environment that weren't previously there? Is it possible that some combination of those factors helped create these fungal pathogens?
I think that's an excellent question, and I'm not exactly sure we still know, what has caused the emergence and spread of this. I think we're pretty confident that human commerce, human transport, shipping of goods from place to place, especially internationally in this globalized world, has certainly increased the volume of trade as well as the speed of trade. And that has really facilitated the spread of these chytrid fungi from Asia to around the world, and probably also many other diseases as well. So definitely humans are involved in the spread of it. What caused the emergence, that still remains to be seen. And again, part of our problem is currently, both of these pathogens are coming out of Korea, China, which are not places where there's a lot of easy access to get information in or out. So this is where things like science diplomacy and international collaborations really become important in building capacity and sharing information across borders to get into the field and figure out, okay, how many other places can we find these things, and what's the genetic signal, and what can we attribute this to?
And has there been any effort globally to try and kind of foster some of that science diplomacy to further investigate and get to the bottom of how these chytrid fungi seem to be originating in Asia? Is there any efforts that have happened since the discovery of this that have further helped that along?
Yes, our colleagues Frank and An at the University of Ghent in the Netherlands, they've been doing a lot of work throughout Asia, Japan, China, Korea, and they have many colleagues there that they're working with. And since they were the ones who originally discovered and described Bsal out of the Netherlands, they really have sort of gone out of their way to really identify collaborators, share information, get materials and information out so that we can get additional cultures which are necessary for the genetic work. Again, it's always a little hard to know what's going on, but the community of scientists who worked on BD originally and have now added Bsal to the mix have been very supportive, very interactive with each other around the globe. And this has been, I think, one of the key lessons that we've learned is that in this world of globalized problems and global threats, it's really important to work internationally and share information so that we can get ahead of these terrible pandemics, whether it's, chytridiomycosis or coronavirus. We are all affected by these terrible pandemics, and if we want to find ways to mitigate or stop them, we're going to have to work together to figure out, what caused them to emerge and what helped them spread, and what can we do to stop that?
Dr. Lips believes a global task force that better understands the impacts of chytridiomycosis on amphibians is crucial. However, current data that is available on these specific effects has recently been called into question. Yet, Dr. Lips concurs that more information is needed, but at the moment still eludes us. I read recently there was a comment on an article on chytridiomycosis that was released in, I believe, March 20th of this year, saying that there was some deficiencies in data about how these fungal infections are threatening species, because currently it appears that it's estimated to be threatening 40% of global populations of amphibians. And I just wanted to know, have you seen that abstract that I'm talking about, and what data deficiencies could there be other than the ones that you've just mentioned?
I have seen that article. I was a co-author on the original paper that Bsal was the leader for. And what I would say is that Ben set up a system, a rubric, if you will, for how to assess impacts of chytrid on amphibian populations. And he initially did it for Australia, where he lives, and published a paper sort of describing the original methods. And then he said, wow, I could use this and in a more systematic way try and assess the impact of chytrid on global amphibians. And what I will do is I will take this same method I have developed and recruit some collaborators from around the world, people who have watched amphibians be impacted or have personal data, and have them go through and assess the impact of both of the chytrids on amphibians around the world. He spent quite some time training people and coordinating this effort, and the paper came out and showed that based on this expert assessment, something like 500 species of amphibians had been impacted by chytrid, and as many as 90 species might have gone extinct. Then, as you mentioned, there was a recent paper criticizing, I guess, the methodology of this approach and basically saying that they didn't always come up with the same conclusions, if I understand it. And what I would say is that Ben had set up a methodology, but all these types of assessments are really based on people's opinions at some level. Now, there were 4 different criteria, and you had to have different amounts of information to assess different levels. But so many of these declines were observed historically or were never observed. And so the original baseline data is incomplete, inconsistent. People's memories and data vary quite a bit in the quality and quantity of information that they have. By nature, then, these assessments, which are very similar to what the IUCN does, the International Union for the Conservation of Nature, they're an international organization sort of tasked with assessing the status of populations and determining if a species is extinct or threatened or endangered or vulnerable, using different criteria. And so Ben's system is not exactly the same, but it is similar in concept to that. But they all rely to some degree on a combination of what are the data that exist, and then in the best of your expert opinion, what would you conclude is the status? I will say that I haven't followed the argument in detail recently, but this is the best we have. So many of these disappearances and declines happened decades ago. We're not getting any new data. And again, what I would say is what we need is really sort of a global task force to get out and resurvey all amphibians and assess how are they doing, how many are there, what are their threats, what are the trends in those populations, because this is what the IUCN Red List is supposed to do, but they rely on scientists to get that information. And it's very difficult to get that information. There's not a lot of funding to just go out and quantify numbers of things. And so as a result, we have very poor baseline data on most of our biodiversity around the world, not just for amphibians, but for all groups.
The importance of this understanding was further emphasized by the fact that we still aren't sure why some Asian salamanders, which carried the fungus, were seemingly unaffected by it. So, in the op-ed that you wrote in the Times, I read that the Asian salamanders that carried Bsal, while they could carry it, they weren't affected by it. What explains that? Have you guys done any research into why they seemed to be carriers but not victims of it?
That's a point that I am still a little curious about myself. Again, I want to emphasize that the amount of information that we have coming out of the wild, that is, information on wild amphibian populations anywhere in Asia, is extremely limited. If it exists, it's probably in Chinese and might not be broadly accessible. But doing a search for information on the distribution of chytrid in Asia and the effects on populations is very hard to find. You can find quite a bit of information. People have been surveying museum collections and have been able to identify the presence of chytrid as long as 150 years ago in, I think it was Vietnam, Thailand, and China, if I recall. So multiple places in Asia have had animals infected with Bcell for over 100 years. Now, what that has done to the populations, I have not found any published articles on that. So when people say that the animals are not affected, I don't actually know if we know that. When you do experiments in the lab with either BD or Bcell, and let's say Bcell with Asian newts, they are highly susceptible. They are very susceptible, they die very quickly, just as American newts die very quickly and are highly susceptible to Bcell. So I think we need a lot more information on exactly how susceptible wild amphibians are to both of these diseases throughout Asia, and to really do some really good demographic studies, population studies of wild amphibians to understand, are they affected? Are they surviving with this disease? Or in fact, is it similar to what we see here in North America or places like Central America today, where the chytrid is present, BD is present, and animals are infected, and oftentimes they no longer live as long as they used to. But because numbers are so much lower today, we do not see big die-offs. We don't see a lot of mortality. So unless you go in and you do a mark recapture study and some good quantitative analysis, you can't actually tell if your population is increasing or decreasing. That is that disease having effect on the population? So I think that's another question that is still open, and that we need field biologists to address that one, just as we need more people in the lab trying to understand the mechanisms of infection.
Even with all this uncertainty around its origins and the effects it has had on certain populations in Asia, we should not forget that the fungus has killed 96% of fire salamanders in Europe, which does not bode well for us here in the US. So, what are the potential downstream effects of a massive die-off? How could that affect us humans? What are the potential impacts that the loss of salamander populations could have on the environment? As I understand it, especially here in the United States where we have such a high concentration of biodiversity of them, I hear that they are actually responsible to some degree in mitigating climate change and carbon emissions. Like, what are the implications? What could happen if we lost a huge chunk of population of salamanders?
We actually know quite a bit about amphibians and ecosystems, but again, there is so much more we need to know. I will tell you that in terms of the effects of BD, the other chytrid on amphibians, we had multiple, I think we had as many as 10 plus years of study where we were working in Panama. And while we were studying the disease ecology of chytrid on frogs, we also were studying, well, when the frogs disappear, what are the impacts on the ecosystem? And so we've published a lot on the multiple roles that amphibians have in an ecosystem. Everything from, you have tadpoles that live in streams or ponds, and they're very important in the food web of streams, not just in the tropics, but also in places like North America and Europe and Asia, where salamanders, frogs, they have aquatic larvae. these little tadpoles and larvae. They feed on aquatic invertebrates. Tadpoles often are grazers of algae. And so when the adult frogs or salamanders disappear from the disease wipes them out, and their abundance goes way, way down. Not only do we lose huge numbers of biomass, like just sheer numbers of frogs and salamanders are way down. And that means that the roles that they play both in the streams and the ponds in terms of regulating aquatic invertebrates like mosquito larvae, or snails, or other small invertebrates, mayflies and stoneflies in those streams, is huge. Plus, you have the added problem of when the adult frogs and salamanders disappear from the forests or the prairies, they also are feeding on adult insects, and they provide food to things like snakes, mammals, birds. And so amphibians sort of occupy a mid-range in most food webs around the world. That is, they're sort of right in the middle, and the larvae are often consuming algae and plant material, and then the adults are really important food. So when you pull out a huge amount of biomass from the very middle of your food web, what happens is you have cascading effects down on the streams, the ponds, the leaf litter in the forest, the insects that are living in these habitats. But you also have cascading effects upwards into the food web, where you have now removed the food of the snakes, the small mammals, the birds that would feed on salamanders and frogs. And so because they are so abundant, this loss from the chytrid is huge and has had these cascading effects throughout the ecosystem. And I suspect we've only really just started identifying what those effects are. So I expect that we'll be seeing a lot more now that people are sort of aware of these cascading effects. Let's see what we can show in terms of the diversity of effects on mosquitoes and flies, and as you said, the decomposition of leaf litter and how that affects carbon and CO2 and climate change.
Because of the potential of these damaging effects, it's frightening to consider the fact that the fungus spread to Europe with relative ease. So I asked Dr. Lips if any study had been done to find species of amphibians here in the U.S. that could possibly carry the disease without being affected by it, therefore making it spread even worse.
Yeah, that's a great question. We've done a series of experiments in our lab, and there's several other labs who are doing all these experiments trying to assess, how susceptible are American salamanders and frogs, for that matter, to this new Bsal, this new salamander chytrid. And what can we expect? Because we're trying to get prepared for a potential pandemic for the salamanders, but also to kind of use this information to prevent it if we can. And what I can say is that based on a whole bunch of different labs working on this, we know that, for example, our red-spotted newt, the eastern newt, which is extremely abundant and extremely common and can be found at really high densities, is very, very susceptible. And there's newts that range basically across the entire eastern half of the U.S. And there's a different set of species of newts in the west, in California and up and down the Pacific coast, that are also highly susceptible, very abundant, high density. And these animals are going to be really important in spreading and amplifying this disease across the U.S. if it gets here. But we also know that there's a bunch of other salamanders, woodland salamanders in a group called the Plethodon. These guys are the little brown or reddish brown animals that you might find under a log in your backyard if you live in sort of the Midwest or the northeastern United States. Many of them are carriers. They may not die, but in lab experiments, if you dose them with a bunch of Bcell, they can be infected. And their infection lasts multiple days, in some cases weeks, and yet they don't die from it. So again, these are these asymptomatic carriers that can move those diseases around, and everything seems fine, but they might be able to spread it quite widely because they don't look like they're sick, and we might not think that they are sick, but in fact they could be spreading disease.
During the conversation, I asked Dr. Lips to tell me one thing about this pandemic that might not be receiving enough attention, And the answer surprised me. Out of everything I've asked so far, is there anything that you wish I was asking that you want people to know about this pandemic?
There is. I mean, I would love it if you could tell people that we here in the U.S., and actually most countries in the world, really we don't have effective ways to keep infected animals out. So we talk a lot recently about pandemics and invasive diseases and closing the border. But yet there is no requirement here in the United States to test any, or I would say most wildlife species to show that they are clean, to show that they are free of invasive emerging diseases like chytrids or coronavirus for that matter. So we have good rules for things like animals used in lab research, so monkeys and mice and rodents. We have good tests for our agricultural animals and plants, and we have good tests and requirements for animals that might be associated with humans, like our dogs and cats and cows and horses. But for wildlife, the frogs, the salamanders, the snakes, many small woodland birds and small mammals, there's not currently any requirement that if somebody wanted to import a bunch of these things, that they would be required to look for diseases or parasites. And so you would bring them in, whether it's for the pet trade or the food trade or what have you. And if these things are infected and get out, they could easily spread these wildlife diseases to many other animals. And as we've just discussed, the loss of wildlife really has big impacts on human health and well-being. You remove all the amphibians and now you have more insects flying around. You've now changed the food supply for birds and mammals. So you would expect to see cascading effects on many of our wildlife and potentially even impacts on human health if we continue to allow infected wildlife animals to come into the United States without being tested. We mentioned this in our op-ed, and there's been efforts. Kirsten Gillibrand in particular has, not this current Congress, but in the two previous ones, introduced a bill that has not gotten very far that would require this type of testing. And it's been introduced both in the House and the Senate. And we're hopeful that given this concern about coronavirus and infectious diseases spreading globally, that this might be a good time and people might be now way more aware of the danger of wildlife disease, because coronavirus started out as a wildlife disease. And so the spread there was through people, but it could have easily have been through infected mammals as well, imports of bats. So this is just to say that we don't know enough that we can afford to keep our borders open to live animals and not take the precautionary measures of looking for infectious diseases. I mean, we're doing it with agricultural animals, so we know it's important. We need to do it now for the wildlife as well.
The United States is one of the largest, if not the largest importer of animals in the entire world. Yet, there is no mandatory requirement to test for most zoonotic diseases. Only a very small handful are actually tested for, and none of them are Bcell, or the coronavirus for that matter. Under the Lacey Act, some 200-odd salamanders are banned from trade specifically because of Bcell, but not all carriers of Bcell are on that list, and as I mentioned, there's no requirement to test for it. And that truly surprised me.
That is exactly what everyone says. I mention this in my talks and in my classes, and every time that's what people, I had no idea that we don't actually do that. And it's true, and it's theoretically we're supposed to, but it's just not getting the bandwidth.
Dr. Lips did mention that any pet store that imports any amphibian that is on that list is supposed to be testing for these fungal pathogens, but that there's no real way to know whether or not they are.
But theoretically, if a pet store was importing anything on that list, they're supposed to get tested first. Are they being tested? Who's testing it? That's a whole nother can of worms that we don't know the answer to.
So that has many scientists believing that the pathogen could show up here any day now. The question is when.
We have no idea. Since our borders have been open and we've been importing millions and millions of amphibians every year for decades, it could already be here, and we just haven't detected it yet. It may be that we got lucky and it's still not here, and maybe the trade is going down and we've put in enough measures that people will be more careful. But when you're dealing with wildlife disease and open borders and invisible diseases, it is very hard to know where is it not, and how to keep it out. Just like, I think this coronavirus is exactly that reason. You walk around and it's like, who's infected? Who might be a carrier? How close can I get? These are exactly the same questions we deal with the amphibians, is which of these things are actually infected? When we go to the pet store, many pet stores are infected with BD. Once it's there, you don't know, are they getting infected once they arrive at an infected pet store, or are they coming in with it with a different type of BD from somewhere else? We didn't even mention the fact that there's now been many other types of BD, different genotypes, slightly different types of genetic variants of the original chytrid that are now around the world that are not here in the U.S. either. And we want to keep those out as well, just like we want to keep Bsal out. So we don't know where these things are. We don't know how they're moving around in the trade because, again, we're kind of blindfolded and we aren't looking at diseases in live animal trade consistently.
Not to add fuel to the fire, but just like different strains of a virus, there can be different variations of these fungi simultaneously in the wild.
Absolutely, and we know that there's what we call these endemic strains. So there's a type of BD from Brazil that is primarily found only in Brazil, but it has been spread in the pet trade to, say, Michigan. We know that there's another one in South Africa that has been spread into Europe and the UK. We know that there's one in Asia that's really closely related to the thing in Brazil. So now we know that there's something like 4 genotypes, we call them. They're different strains of the BD chytrid. And so you might expect to find different strains of Bsal perhaps as well once we get out there and start looking. So we definitely have not completely identified the diversity of chytrid fungi in the world. And so that's yet another thing that we need a lot more research to figure out what's going on.
Dr. Lips provided an enormous amount of insight, some of it hopeful, much of it worrying, about not just the threat of Bsal, but the threat of wildlife diseases. Needless to say, her expertise is heartily appreciated, but I particularly found her parting words to be the most poignant.
I'd like to thank you for covering this. I think it's incredibly important. I'm hoping that this coronavirus epidemic will raise the awareness of wildlife disease in general to our citizens, and that people will realize that we are all connected, and healthy environments have healthy animals, and that's good for people, and disrupting the environment can have very many unintended, invisible, cascading effects on humans as well. So we need to do a much better job of protecting our environment and living sustainably.
I don't think I could have put it in better words. Thank you all so much for joining me today. Please join me again for our next episode, where I will be speaking with Mongabay staff reporter Morgan Erickson Davis on what makes the United States the global hotspot of salamander diversity, and how this unique situation plays into this potential pandemic. I want to thank Dr. Karen Lips for her time and her service to the global scientific community. And also a special thanks to Rhett Butler and Morgan Erickson Davis, for whom without this podcast would not be possible. Watch for a new edition of Mongabay Explores every 2 weeks, in between episodes of our flagship podcast, the Mongabay Newscast. Special projects like this are made possible by our Patreon supporters. Mongabay is a nonprofit news provider, so we rely on the generosity of our listeners, readers, and friends. To add your support, head to patreon.com/Mongabay to learn more. Keep up with all of Mongabay's news from Nature's Frontline at mongabay.com, or get updates via Twitter, Facebook, and Instagram, where our handle is @Mongabay. Thank you once again, and we'll be back soon with another episode of Mongabay Explores.